Good evening to everybody, and welcome to the first lecture of the 2025 edition of Horizons in Medicine and Biology. I am Lorenzo della Casa, and this is my colleague Alessio Morosi, both second-year medical students here at Scuola Superiore Sant'Anna. Before we begin, here's a brief introduction for those unfamiliar with this institution. The Scuola Superiore Sant'Anna is a public university that admits students purely on merit through a highly competitive national selective process. Students at the school have the opportunity to tailor their university studies to their personal interests. They thrive in a dynamic, multidisciplinary environment where individuals from different fields come together to participate in shared courses and projects while living in the same community. This collaborative setting fosters the exchange of ideas and nurtures innovation.
Thanks to this event, Scuola Superiore Sant'Anna is thrilled to welcome distinguished experts in the biomedical field as one of us for a day. The host will join our community, sharing his expertise and journey with students and faculty alike. This unique opportunity to connect with such inspiring figures underscores the school's dedication to creating moments of meaningful interaction and inspiration. Students will gain invaluable insights not only into the latest advancements in medicine, but also in the professional path of leaders in the field. For one day, they become a part of our academic family, leaving a lasting impact and sparking new ideas that could shape the future careers of many. The rich cultural environment led to the establishment of this lecture series. These seminars give us the opportunity to host in our school many brilliant researchers devoted to medicine, who gave us a little pieces of their great research experience. Before the start of this lecture, we would like to share with you a short summary of past editions of Horizon in Medicine and Biology through this video.
Students like yourself are very impressive to me, and I can understand that it is a very strong selection.
La Scuola Superiore Sant'Anna è ovviamente accompagnata dalla fama dell'aver formato tante persone che ricoprono ruoli importanti nel Paese.
That's something very special, and something very exclusive and unique, and so immediately I said, I want to be there, I want to do this lecture, and I want to know the people there, how they work, because for me it was a surprise.
Per esserci stato in passato, ma anche per fama, perché la Scuola di Sant'Anna è certamente una delle istituzioni educative fra le migliori in Italia.
Orizzonti mi permette di conoscere e approfondire quelle che sono le ultimissime innovazioni nel campo della ricerca biomedica e di conoscere da vicino i protagonisti che fanno.
Orizzonti mi permette di esplorare temi che non sempre si affrontano durante i percorsi universitari e di capire come la ricerca e l'innovazione possono tradursi nella pratica, cosa che sarà fondamentale durante il mio percorso lavorativo.
Orizzonti significa per me futuro, ricerca e scoperta.
Internazionalità, cura e conoscenza.
Thanking Professor Soranzo for being with us today, I give the floor to Professor Vincenzo Leonetti, the architect of this seminar, who will introduce our guest. Thank you for all.
Good evening and welcome to this new great lecture that Professor Soranzo will have here today. Just some information about our guest. Nicole Soranzo is actually the head of the Genomic Research Center, the Human Technopole in Milan, and she's also been a senior group leader at the Wellcome Sanger Institute since 2017, and she actually an associate member of the faculty and professor of human genetics at the School of Clinical Medicine, University of Cambridge, since 2015. But as we know, Nicole Soranzo is Italian, is an Italian scientist, and after completing her degree in biological science at the University of Milan in 1984, she obtained her PhD in genetics and biotechnology at the University of Dundee. From 1999 to 2002, she carried out a postdoctoral research at the University of Milan, and from 2002 to 2005 at the University College in London, where she applied human genetics to study human evolution. From 2005 to 2007, she worked as senior scientist at the pharmacogenomics department at Johnson & Johnson Pharmaceutical, applying human genetics to improve drugs discovery in pharmacogenomics. She returned to the UK at the Sanger Institute, where she started her group in 2009, and in 2013 she became adjunct faculty at the University of Cambridge School of Clinical Medicine, and in 2015 she was awarded the personal chair in human genetics. For her outstanding performance, she received many awards and honors as the Italian female researcher and scientist of impact, National Observatory for Women Health. In 2016, Nicole Soranzo studied how the human genome influences the risk of common disease in the general population. And it's just a summary because the CV is much more long. And for me, it's a great honor to have here on the podium Professor Nicole Soranzo for her lecture. Thank you.
Thank you. I really had a lot of fun today, and it's really been a pleasure to know your school, you know, your reality, and what you're doing here. I was familiar from a distance, but it's another thing to sit up close. So I'm a geneticist, so this is the Sanger Institute, where I spent the majority of my really formative years as a group leader, and decided recently, a few years ago now, to move back to Italy. Not because I missed Italy, but because I wanted to be part of the setting up of this new Life Science Institute in Milano, Human Technopole. And I'll give you a little bit of information about the Institute later, because I think it's also important to understand what's happening and to connect to the new reality in Milano. We worked for now many years on common chronic diseases, particularly chronic diseases of old age. And as you all very familiar from your medicine degree, these are the biggest killers, biggest causes of mortality, but also of life lost to disability and ill health worldwide. And cardiovascular diseases are the top killer together with cancer. And they're very much an Italian problem too, because the Italian population is aging very rapidly. We estimated by 2028 there will be 25 million chronic patients in Italy, and this results in a huge expenditure for the public health system, estimated at 67 billion euros per year. Progressively, the problem is getting worse. Italy is the country with the highest aging index in Europe, where the elderly have already outnumbered the young 2 to 1 a few years ago. this is not just an Italian trend, this is a worldwide trend that the so-called non-communicable diseases are indeed growing. And this is a combination of it's not of course due to genetic changes, but rather to changes in environments and lifestyles that are major contributors to predisposition to these diseases. And what makes it quite difficult to study these diseases from a genetic but also molecular view is that typically these diseases, especially in old age, co-occur in multimorbidity condition where you have essentially effects of aging, progressive deterioration. So this is a case of immune system that we focus on, including the onset of inflammation. But this is also due to environmental stressors such as infection or stress itself, but also, as I said, to the presence of comorbidities such as, for instance, the joint influence of obesity, diabetes, immune failure, but also hypertension. So this makes it quite easy, quite difficult to study. So when we study complex diseases, both genetics and otherwise, we always have to take into account these different components. So genetics is one of them, but also aging, lifestyle, and environments. And so this is, you can think of a person as essentially a bucket, each person carrying a bucket of risk. The blue dots are your, individual units of risk that come from your genetic risk, which is inherited. And then, of course, the red is the age, which, of course, interests all of us over time. And then different environmental and lifestyle choices. And so, for instance, you could get to a position where you develop disease because you have a high joint risk of genetics and environmental risk. Or you could indeed become sick simply because by the unlucky draw of the lottery you have inherited many predisposing factors to disease. So you have a high genetic burden for that particular disease. So we've been thinking really hard about how do we apply our new technological abilities, both in genomics but also in other disciplines, to narrow down the causes of chronic human diseases. What we do in particular, as many members of the community, is to use genetics. And we use genetics both because this is as a cause, but also essentially as a tool that allows us to dissect biology, to resolve causal effects, and to define the interplay of intrinsic but also extrinsic factors that affect each individual person. And so this is perhaps the premise of what we call personalized or precision medicine more generically. So as I said, much of our research and the ability to do cutting-edge research depends on cutting-edge technology, and this is particularly true for complex disease studies. So in the last 20 years, we saw a huge revolution in really a paradigm shift in our ability to study genetic causes of disease, and this has been given to two main factors. On one side, the sequencing of the human genome that essentially provided us with a blueprint where we can do searches in silico for things that would take normally years to complete. And the other is also the development in sequencing technologies that made it cheaper and cheaper to sequence genome on a large scale. And so if we know that the advent of next generation technologies, which are no longer next generation, we have essentially seen a huge decrease in the cost of genomes. That means that this approach has been, we have been able to apply this approach to thousands and thousands of people. So scale is hugely important. And so if you're really interested in understanding a little bit what the progress of this revolution has been in the last 20 years or so, I would point you to this review that I wrote together with many colleagues in my field. And this is essentially show the progress on the left of the discovery of genes that underpin Mendelian disorders, and on the right, the discovery of variants or polymorphisms that predispose you to complex disease risk. And so you can see that on the left-hand side, my pointer is not working, but essentially the left-hand side, from 2010 onwards, when I set up my group, we had a huge increase in knowledge, and this is also partly through the efforts of my group at the Sanger Institute. And so we have seen essentially the creation of what we call a virtuous cycle, where we have new genomic technologies that drive the accrual of more and more data, that drive scientific knowledge, that in turn drives the development of bigger and bigger data set, and that drive the development of new technologies. And this is a cycle that doesn't include only genome sequencing, but progressively other types of genomic technologies, for instance, single-cell RNA sequencing that you hear next week about, and many others. The cornerstone of how we do genetics is using very large-scale human population studies. So essentially, we use a person like an experimental system, like an in vivo laboratory. And we choose this because we want to have a view of conditions or pathophysiological elements of disease not in isolation, but in conjunction with essentially this system view. So typically population, and this is an example of the UK Biobank that has really driven worldwide research, but they involve participants that are predominantly healthy, typically unselected or representative of the population of origin. They have typically extensive phenotype information that comes from profiling of risk factors, so traditional risk factors such as, for instance, a level of bad cholesterol or glycemia, but also progressively more types of omic data that provide candidate new biomarkers. And more recently, imaging data is becoming a huge driver of scientific progress. And then we have, of course, we want these data sets to give us information on disease that typically come from self-report, but also from progressively more hospital discharge record or primary practitioners record, and from measurement of environmental exposures, including data from questionnaire, but also objective measurements such as physical activity or neurocognitive tests, so just to mention a few. As I said, UK Biobank is a UK resource that has really pioneered the utility of doing this type of research. It's a research capability that has some fundamental underpinnings that, in fact, drive the use and the resource. Is first of all large, includes 500,000 people, and this provides you with the statistical power that you need to do not just small lookups, but really very powerful statistical inference with a good understanding of the causal relationship between, for instance, risk factors and disease. It is highly standardized, so the data is curated to an incredible degree, and this allows you to essentially reduce statistical noise, but also to be able to compare across all the different types of data modalities that are present, and very high quality. And complete. One of the decisions that has been made in the recruitment of 500,000 people is that every phenotype that was measured in this cohort would be measured in everybody. And this, again, provides you with a platform that you can use for statistical analysis. And the key point is that this resource has been designed to be shared with the worldwide community. So if you are a scientific researcher, no matter where you work or where you come from, you can use the data through, of course, the appropriate legislative measure using control access. And this is an important component of policies that in the UK are used to democratize investment and increase reuse of data. And this is perhaps one of the points where in Italy we're really still struggling, is to allow the secondary reuse of data, for instance, clinical data for research. But it's a hugely powerful position, philosophical position. The way we tackle genetic studies has been, again, as I said, through improvements in knowledge and understanding of biodata. And one of the key advancements that we made before genome sequencing was sufficiently cheap was to understand that in human population, DNA variation is inherited in blocks, what we call linkage disequilibrium. So essentially, each of these red or orange line in the plot before is what we call an haplotype, and the variants within that haplotype are inherited in block. And so what this discovery and the characterization of this haplotype structure in different populations allowed us to design low representation arrays, within this called SNP arrays, that were cost effective and provided us with the first opportunity to essentially serve a genome-wide variation in population when sequencing was too expensive, and in fact when next generation sequencing hadn't been yet invented. So we use this type of approach initially for association studies. The other type of mindset that we use, as I said, talking about the importance of phenotype, is that essentially any human trait that is measured can be considered a useful phenotype. So say if you want to study cardiovascular disease, you could take a collection of people that are healthy and a collection of people that had a myocardial infarction, and you could look at genetic differences between them. But you can also do other things. So for instance, you can take quantitative measurement of the arterial narrowing as a quantitative trait, or indeed molecular, very well-defined molecular processes, for instance, here involving the development of the atherosclerotic plaque through progressive onset of inflammation. And the advantage of that is essentially you can break down complex phenotypes, such as a heart attack, into the individual subcomponent and study how individual genetic variants or genes contribute to individual components of the pathophysiology. In general, this genetic association framework proved to be extremely powerful, not just because of the statistical power, but also because of the underlying genetic assumption. And that's the one highlighted here. So we started these association studies without any a priori belief of where the causal variant, where the genetic effect is. We had a completely agnostic view of what genetic variant or gene cause disease. And this allows us to essentially go without preconception and really look at the evidence that it gave us. Of course, it's also a very cost-effective way when you have cohorts that are phenotypes for thousands of phenotypes. So we've been implicated in the early wave of genome-wide association studies using this type of approach. And these are just some of the publications, these are now old work, but essentially we worked with continuing to enhance the genetic characterization of our samples, but also initially looking at disease risk or, in fact, risk factors. Progressively, what we realized is that in other types of phenotypes, such as I said, molecular phenotypes, provide a very useful way to dissect this relationship. And so, for instance, we looked at gene regulation phenotypes, so essentially gene expression measured in primary immune cells in people drawn from populations. We looked at protein levels, again, quantitated from plasma or serum from thousands of people. We looked at metabolites, which are essentially small molecule components of protein, and even more so, we looked at cellular phenotypes, such as, for instance, those derived from clinical blood counts. And all of these components essentially prove to have some, general characteristics. So actually, most of these phenotypes are at least partially heritable. So we could do genetic association studies on them, and we were able to define genetic control at every layer of biology. As you move from the left to the right of the spectrum, you see that traits become more and more complex. So few variants control, for instance, the expression of a single gene, but hundreds of variants control expression of genes implicated in myocardial infarction. And so this simplicity at the left-hand side of the spectrum allows us, as I said, to break down complex phenotypes into the individual component. And then the other thing that became very obvious is that there was a huge genetic correlation between the genetic effects on these molecular phenotypes and the genetic effects on disease risk. So essentially, we can use this approach to pinpoint which genes, which molecular processes, which, for instance, methylation sites or RNA changes are implicated with the pathophysiology of different diseases. And this is just to give a sense for how quickly we went. In 2005, we had discovered the first common locus underpinning a complex disease predisposition. As this progress became bigger over time, we started essentially filling the entire genome with predisposition factor for every single biomedical trait that you can think of. And this not only gave us a good catalog of genetic variation to work on, but also allowed us to pinpoint cases where, for instance, diseases shared a single genetic cause. For instance, explaining cases of comorbidity of different diseases or the possibility of identifying therapeutic targets through shared genetic effects. We learned a lot also about, more what we call the allelic architecture of complex diseases. So whether, for instance, what is the distribution of risk factor for diseases. And so, for instance, the variants that we discovered to these initial type of approaches, we found out tended to be really common in the population. So they had a high allele frequency in any population. They were often located outside protein-coding regions, so they presume to act through regulatory rather than non-coding differences. Indeed, individually, they have very small effect sizes, meaning that each genetic variant has a very, very minuscule effect on a disease. And this is really in opposition with variants on the other side of the spectrum, for instance, Mendelian variants, but also other types of variants that we're now discovering through population sequencing that are individually very rare, but contribute in the person that carries them a high chance of genetic risk. And so part of how we are now thinking about the use of genetic variation, for instance, in prevention or in prediction, is really driven by our understanding of disease, of this kind of allelic architecture. The other thing that becomes very obvious is that in order to develop a complex disease through genetic risk, you have to have many, many, as I said, elements of the genetic risk, high burden, as I told you before. So essentially, if you think about, for instance, identifying all the variants associated with myocardial infarction, typically people that have a lot of these variants through their genetic lottery, they tend to be a higher genetic risk for disease compared to people that are on the left-hand side of the spectrum and are protected. And I'll tell you later how we're using this information. So as I said, this field has progressed incredibly fast. We have now commoditized completely the use of genomics in populations. There's now millions of samples being sequenced all over the world. And what this is giving us is an opportunity to essentially look up very quickly in silico information in public databases. This is really cutting very short the time of data to experimentation. We're now at the stage where we have accrued probably as much knowledge as we could accrue in terms of these genetic risk factors, at least in the type of mapping studies that we're doing. And so now the challenge becomes that of understanding how we can better use this information for clinical translation, essentially to accelerate the prediction and the treatment of chronic kidney diseases. And I see this as really an effort of essentially gathering all the information that we have, not just on genome sequencing, but also on functional genomic data sets and leveraging new technologies such as machine learning to essentially come up with new hypotheses for treatment. And genomics is really, as I said, revolutionizing everything what we do in terms of discovery of human disease. As you've seen, provided us with information on biology of processes in health and disease. Is helping us understand the causes of all diseases, the interaction with different environments, so we call this disease etiology, but progressively more is helping us inform the management of health through the development of risk prediction and early detection algorithm, as well as the development of new drugs and tailored treatment with therapeutic targeting. So I want to give you an example of these last two applications, because you are medical students, but I want to draw mostly from our experience at Human Technopole, so just to give you a sense of the kind of studies that we're trying to do in Italy, and these are mirror to some extent the kind of studies that we've done before. I'll talk later about Human Technopole, but just to see that we are a genomic and life science institute focused very much on technology, but we're very embedded in the national landscape we're working very closely with population cohorts, with the healthcare systems, with entities that are already using quantitative data science faculties, such as statistics and mathematics, and very much the Polytechnic, and also progressively more with the pharmaceutical industry. And, while Italy has, some underinvestment in research, we do have a superb collection of clinical samples, superb clinical expertise, and the wealth of a national and regional healthcare system that can be harnessed for this type of studies. So when I joined Human Technopole, we set a number of goals that we're trying to put forward. First of all, to develop population resources that could comprehensively capture Italian genetic variation. The second was to assess the burden of genetic variation on health and disease, similarly to what we've already done in the UK. To develop algorithms that allow us to assess the long-term consequences of genetic risk in Italy, and to assess the impact of unique environments. I just want to take you through some of the kind of studies that we're doing at the moment. So the first one is really, as I said, stems from the fact that we have a very incomplete view of predisposition in Italy. And if you look at essentially the representation of the fraction of the population of each European country that is currently sequence or genotype, we are really at the bottom of the scale. We are one of the countries with really the least genomic data. And this is a problem because, of course, this prevents to some extent the use of genomics in the health system. And it's particularly important to study Italy by itself because, as you all know, Italy is a genetic patchwork. We sit somewhere between with the Southern European countries, but we had multiple waves of invasion, colonization, migration. We have a lot of genetic isolation through our unique geography and history. And as a result of it, the degree of population differences within Italy, which is this violin plot shows on the right, is as big as the genetic diversity of all Europe combined. So we have a lot of heterogeneity that we need to deal with. And this is not just an academic problem, but it's also a medical problem, because the moment you see a patient in a clinic and a patient has a genetic mutation, you need to make a call on whether the genetic mutation is common in the population, and therefore that the finding is of no clinical significance, or indeed is very rare, and that might be the driver mutation for your disease. So characterizing population structure from a genetic point of view is clearly important for medicine too. So we have set the goal to generate, now whole genome sequencing in tens of thousands of individuals that are representative of all European regions. So we've done essentially a first effort to collect historical data to essentially map, create an anchor, a grid of genetic variation across Italy. And we overlay onto this grid then an aggregation of existing genomic data sets to understand the fine-grained structure of human diversity, and to apply that in the clinic. And for this, we have now generated collaboration with multiple scientists from all over Italy, and we are now part of a very large-scale European grant called the Genomes of Europe, which is a large infrastructural grant that seeks to extend this approach not just to Italy, but indeed to all European countries. And so the idea is that the whole of Europe also will sequence 500,000 genomes across all of Europe with proportional representation of different countries. And the idea is that if you are a clinician that is treating an Italian patient in Belgium, you can draw to this collective knowledge to do that kind of process of inference of genetic risk. We're also doing some more, if you want, modern or cool genomics, and this is essentially work by Davide and Alessandro in my team that essentially is called pangenomics, which is essentially to revert our traditional view of a genome as a linear entity and really harness the complexity of the genome, a new computational algorithm to harness the complexity of the genome. And I just put this here because this is really cool work, but I won't be able to talk about that. The second really key function, point is that we also want to, similar to what I told you before, try to develop tools that allow us to assess the burden of genetic variation in the Italian population. I won't comment this further, but this is again a snapshot of worldwide cohorts that are being generated, or large size. As you see, Italy is in the non-existent space, but what you also see is that there are substantial investment in other countries that you would think unlikely, like Brazil or South Africa, Iran or Saudi Arabia, that are already developing genomic strategies. So we're not just behind the developed world, but progressively even more the developing world in this sense. So what we want to do here is a mini UK biobank. So the idea is to collect information from tens of, in fact, we were aiming hundreds of thousands of volunteers all across Italy to capture essentially a local, not just genetic risk, but also local environment. So to have a fair assessment of the local exposures. For instance, exposures that are linked to differences in diet or fitness regime. And so we set to do this. Of course, this is a big challenge. We set to do this through a combination of setting up resources and collaborating with existing ones. And one actually of the incredible resources that we discovered in doing so is a so-called the Molisani study, which is essentially a prospective cohort that was set up nearly 20 years ago in a very visionary way by colleagues shown here. So this was set up as an epidemiological cohort, essentially a survey that was designed to understand whether, what were the local risk or protective factors, excluding genetics, environmental, biomolecular, that are linked to chronic degenerative diseases in the Molise region. So why Molise? It's a very restricted geographical area with close to no immigration, and a typical southern lifestyle. And what made this study possible here is the fact that there was a really close connection between the scientific community and the political community that allowed them essentially to operate in a highly synergistic way to create this incredible resource. And I've been told that Molise doesn't exist. It is a legend, but I can attest that it does exist. It's a wonderful place. So in this case, what they did is essentially they asked essentially GPs to recruit randomly people drawn from the registry office. So in this way, they knew that there was no bias in terms of the person that were invited to participate to the study. There are two main recruitment centers on the other side of Molise. Campobasso is in the mountains and Termoli is on the sea, but they involved 30 municipalities and involved nearly 25,000 individuals. This is an incredible size of the study for the Italian cohort. And what they did is that this happened between 2005 and 2010. They not only collected DNA, but in fact they collected information from a huge biobank, including blood, plasma, EDTA plasma, serum, cellular pellets, urine, and so on. They recruited individuals that were willing to donate their time to collect a wide host of biomedical traits, including many risk factors, and agreed to progressively give information over time. And now we have nearly 20 years of follow-up, meaning that we can follow whether a person dies or gets ill by any disease through linkage to electronic health records for that person. And so we designed a SNP array that allows to study this kind of haplotype tagging variation, but also variation that are overrepresented in Italy and variants of medical interest. And we now done a study linking this genetic variation to a very wide range of not just quantitative risk factors, but also disease diagnosis. And we're now finding essentially what is the allelic architecture of complex disease risk in Italy. I will not go too much in detail, but what we can see is that there are very strong similarities with Northern European findings, but we're also finding variants that are unique to Italy. And this is one example. So this is a plot that shows the association in a region of chromosome 11 that contains the HBB, the hemoglobin beta locus. As you see, there's a huge red peak in Molisani, but no concurrent peak in UK Biobank. And this is essentially corresponding to the beta-globin gene cluster, and what we see is that there are indeed variants that are being associated with minor thalassemia that show up as nearly uniquely Italian. They've also been found one in Greece and the other in Sardinia. So it's very clear that for the individuals that carry this mutation, genetic information is incredibly important. So how are we applying the kind of genetic information that we developed in these cohorts for prediction? So we use what is called the polygenic risk score. So these are essentially becoming mainstay information. And the basic idea is very simple. So we take each person. We now have large catalogs of genetic risk for any essentially quantitative trait risk factor or any risk factors, and we simply add up essentially the number of risk allele that each person carries, so we can characterize people that are high or low risk of disease. And we essentially weight the sum of this genetic information in a polygenic risk score, which is a quantitative trait. And one of the advantages, so there's a lot of now debate on the utility of the polygenic risk scores in a clinical practice, whether they're useful or not. And the main difference from non-genetic risk factors is that they provide you with a stable risk assessment throughout a lifetime that is not modified by disease. So it provides us with an incredibly powerful tool. And in fact, it's been shown that polygenic risk scores are useful for improved population screening of diseases, so such as the Bodicea algorithm for breast cancer. They are useful for the estimation of risk trajectories across lifetime and for the enhancement of disease risk prediction and stratification for many different diseases. So much so that clinical trials are now being done to incorporate polygenic risk scores among the risk factors that are assessed. And this is one example. So this is essentially a company in the UK that is develop polygenic risk score predictors, and essentially individuals then enter clinical trials when they are randomized, not just by, for instance, the family history or the level of cholesterol, but also by the genetic risk. And this is shown to improve essentially the identification of successful drug targets. One of the problems with the polygenic risk scores is that these are typically discovered in population of northern ancestry, and of course we don't know how well they work in populations that are genetically different, such as, for instance, Italy. So there's been shown that there is clearly a decline of predictivity that is correlated inversely with the genetic distance between the original population and the target population across ancestries. The question is how much of this loss of predictivity happens also within Europe, and in particular between Northern and Southern Europe, and this is what we set to investigate. So we set up, essentially, we used our Molisani study. We set up a highly controlled experiment that I will tell you about. But essentially what we find that polygenic risk scores are useful, clinically useful in Italy. And this is just one example. So here we have plotting the cumulative hazard ratio of developing a cardiovascular event over the time of follow-up of the Molisani study for people that are stratified based on whether they are in the middle category of risk, so they have an average risk, or people that are in the extreme left-hand tail or top tail. And so what you see is that there is a twofold difference of developing coronary artery disease for the high-risk category compared to the low-risk category, indicating that genetic stratification in this case is incredibly clinically useful. And we now have different types of algorithms that allow us to compare the clinical utility of adding genetic information on top of clinically used scores, such as, for instance, the score 2 for cardiovascular disease, or on top of family history alone. And we see an increase, small but significant increase in predictivity by adding genetics. Another case where there is a clear advantage in using genetics is for the population screening of breast cancer. So in Molisani, we have 470 breast cancer events, If you see, these are unevenly distributed between categories of genetic risk. People that are in the blue line, so the low risk tail, they have only 88 events over the follow-up time, whereas people in the high category, they have 248 over the same period. So essentially, what you see is that half of the breast cancer cases are observed only in the top risk category. So we ask whether we can use this information to come to some information about whether incorporated genetic risk can help us tailor in more efficient way the population screening of breast cancer. So this is a zoomed up plot of the same distribution of the cumulative risk of developing breast cancer over time. So at age 50, that's typically when a woman in Italy would begin their mammographic screening. What you see is that at 50, about 0.4% of people develop breast cancer if they are in the low-risk blue group. However, the same proportion of people, in the same proportion, is achieved at age 40 for people that are in the top risk group. So essentially, if you want to achieve the same clinical benefit, you would not need to screen the people that are in the top category of risk 10 years earlier than others. And so this really opens a possibility that genetics can make public health screening more effective. And so potentially, so by suggesting that the people that are unknown higher genetic risk should be screened earlier. And so we estimated what would be the benefit of changing the screening strategy informed by genetics, and so you would detect essentially a quarter more cases out of the 2048 event that we do. So it's a big difference. We also use the same type of approach, also look at causes of mortality, and in fact, what are the highest determinants of mortality in Molise, and this is just an example. So here we are comparing people that are high risk of genetic obesity, whether they're obese or not, but they have a high genetic risk versus people that are low genetic risk, and we see that there is indeed a 30% increased risk of mortality over a 15-year period if you are a high genetic risk of obesity compared if not, and this corresponds to a 2-year difference in life lost. So as I said, the benefits of genetic is that this prediction is easier because you can predict, you don't have to measure BMI at single time points, you measure genetic once, but importantly, this knowledge is available at birth, and therefore you can tailor, again, screening or preventative measures in a more efficient way. Okay, the last point is that we're also trying to do is to assess impact of unique environments, and this is because, of course, Molise is not representative of the Italian population at large, and this is just an example of the fact that if you take any cardiometabolic risk factor over age brackets in Molise and you compare that to other Progetto Cuore, in this case, which is an Italian-wide population screening program, you see that indeed the risk profile is very different. Molise population is a higher risk of cardiometabolic disease based on their risk factors, and this is why we want to extend this type of approach to representative realities throughout Italy. Okay, the last point where genomics is really making a difference is in the therapeutic targeting. So the idea that we can use genomics to inform in more efficient ways how we develop and discover drugs. And I think you're all familiar with the complexity at the moment of developing drugs, and in fact the huge attrition rate of target identification throughout pharma pipelines. So what has become very clear from this type of genetic studies is that if you have a genetic evidence that links a genetic variant to a gene to a disease, that increases tremendously the chance of developing an effective drug. So what we've done here is essentially, of course, we haven't had yet the time to develop new drugs from genetic data, but what we can do is to go retrospectively in drugs that are already approved by the FDA and ask what is the chance that a drug was successful depending on whether they had genetic information or not. And what we see is that the odds ratio are very high, in the order of at least twice more efficient, sometimes up to 40 times more efficient to develop a drug using this approach than not. So essentially what we are doing here is to now developing algorithms that allow us to mine through all the big data sets that we generated to link a SNP to a disease and to a risk factor. So we can ask, does a genetic variant correlate with the modifiable disease risk factor? And does the risk factor causally contribute to the risk of disease? And then we can link the genetic variant to the gene and to the drug and asking whether the genetic variant acts through that particular gene, and is that particular gene druggable? And essentially what we do is use this type of information. We look for genetic variation in patients and families. We use different types of functional genomics approaches to link genetic variation to genes. We test for causality and direction using statistical approaches, and then we go back in population and cohorts. And I will say that, I think there is often a sense that pharma or, big pharma and research really don't coexist, or they are driven by different sets of ethical values. But having worked in pharma, I can tell you that this is certainly not the case. There has never been a time as now where science, academia and pharma are indeed working cooperatively together. And so, for instance, one of the pharma companies, Regeneron, is really taking genomic research to industrial scale. They are sequencing now multiple millions of sequence and of genomes across the world, and they are now, developing new drugs for indications that we didn't know could be indeed tackled. And this is done not just through genetics, but of course through big data analytics and functional genomics data sets. Okay, I'll skip through some of the other things, just to say that this kind of science can happen through collaborative endeavors, but is also enabled through private partnerships, so where essentially governments, academia collaborates with pharma, and this is very much the model that we hope to implement. Okay, I'll skip through because I don't want to be too late. And just maybe a touch on where we're going in general, and I think you'll hear a lot more about this next week. But essentially, we are really, we're entered in what we call the post-genome era. So we are very much done with sequencing people, other than perhaps in some realities. And we're really working to, the next stage, which is the stage of interpretation of functional annotation, and really understanding more of the complexity, biological complexity and environmental complexity that genetic variants act within. So you'll hear next week about how single-cell technology, for instance, are revolutionizing the use, the interpretation of genetic variation from Sarah. Just to say that we are also now exploiting the fact that these technologies are being scalable to apply the same type of data also to population cohorts. So we have a big grant by the Chan Zuckerberg Initiative that is allowing us to do that. And so the idea here is that we take individuals from cohorts with biomedical data, we profile them extensively with single-cell profiling for circulating immune cells, and this idea is that we look at assays that are scalable, they're accessible, they provide a definition of immune state and also provide information on the etiology of chronic diseases that have at least inflammatory immune-mediated components. But of course, this coming from circulating blood allows us also to great potential for clinical implementation, for instance, in the development of disease biomarkers. And of course, this is important because it gives us insights into age-associated decline in protective immunity, which is linked to the risk of many, many chronic diseases. And it provides us with huge translational value in terms of developing markers for disease diagnosis, using drug signature for biomarkers and drug discovery and repositioning. And I just want to say that we're using similar approaches to what I've shown you before to link variation in immune cell subsets at incredible resolution with many immune diseases and not just immune diseases. And we are now identifying thousands of human genetic variants that have an effect on disease through immune components. And as I've shown you before, this is an approach that allows to very quickly identify and nominate drug targets. So, for instance, the odds ratio of developing a successful preclinical drug is 8 when you use information from these single-cell technologies. And in fact, becomes astonishingly high for essentially predicting whether a drug will be successful in moving from preclinical to phase 1, phase 2, and phase 3 stages. Okay, other types of innovation that we're using are machine learning. We're very much now moving into the era where we don't analyze genetic variation one by one or phenotype one by one, where we're trying to make sense of complete data sets with machine learning algorithms such as large language models or decoders. And in fact, this allows to model in a much more sophisticated and potentially realistic way the interaction between genetics, disease, environments, medication, and so on. And so, of course, this opens a new era where we can harness not just genomes, but indeed post-genomic analysis to continue to do prevention, to develop new therapies, to get biological insights, and progressively more to understand, by leveraging population differences, causes that are specific to different populations and to promote equity. I'll give the last 5 minutes, if I have, to tell you a little bit about what we've done in Milano, because I think it's important, and this is really to extend also an invitation for you to come and visit us and work with us. So, as I said, I moved to Milano in 2022, started working with them in 2020, and really the idea was to develop a new life science institute that could work as essentially a multidisciplinary institute and an open hub of technology for Italian community. So this is our headquarters, which is the Italian pavilion at the Expo in 2015, and the whole project was funded at the tune of 140 million per year by the Italian government, really as a way to build a long-lasting legacy to the success of Expo and what it brought, particularly to Milano. So we set up a large-scale infrastructure, multidisciplinary laboratories and research facilities for the development of precision medicine, but this is intended in a very, very broad sense. And in the space of only 4 years, we were able to recruit already 26 different research group leaders and to establish 6 different research centers. And while we're now working still from temporary labs, we have now a plan. In fact, we're starting the planning of this big building on the left-hand side, which is essentially a 10-story building that will host the institute, but will also host Italian community for periods of time, so an opportunity to visit us. And just to look at the timeline, essentially just to say this was really a lot of work in a very limited amount of time. As of December of last year, we have recruited nearly 500 staff, and we really try to make the institute as international as possible. We speak English, we have 39 different nationalities, 370 research personnel, and we have already raised over 26 million of external funds. So I'll skip to that. We have 2 components of, if you want, 2 souls to this institute. One is what we call the research component, and this is really a highly multidisciplinary endeavor where we have essentially technological capability and expertise that allow us to look at, essentially to explore biology all the way from the structure of molecules in cryo-EM type settings, all the way to population genomics and population health, with everything in between. And so this really puts under the same roof a very multidisciplinary set of expertise, but also a technical capability. And so we went and essentially mapped where each research center kind of fits, and as you see, there's very much potential for crossover and contamination between all of us. I will not go too much into detail of the institute, but just to highlight neurogenomics does a lot of work in brain organoids, computational biology does a lot of work on cancer, and genomics does a lot of work on fundamental mechanism for immune-mediated and cardiometabolic diseases, and so on. Our center has now 6 PIs, and one of the PIs is actually from the University of Parma, Elisa Araldi, and she's a graduate of the Normale School in Pisa, so very much local talent. So we set up individual research labs, but we set up in a facility. And essentially what we're trying to do is to build approaches that allow us to go all the way from population genomic approaches that help us discover and predict and treat human diseases, and this is what you heard from me, but also all the way to developing cellular functional models that allow us to get insights into mechanistic and regulatory control, particularly of immune function, to develop cell-based models that we can perturb to develop drugs and improve patient outcomes, and the development of new algorithms. So, for instance, Craig Glastonbury has a lab that uses very much the development of new machine learning method to analyze different modalities of imaging data to essentially do in silico histopathology and genetic association. And the Giuseppini lab works on vulnerabilities in leukemias. The Dominguez Conde lab works very much on immunogenomics and development of the immune system and impact in rare disease. And Soskich lab in immunogenomics and the development mechanism of antibody diversification. We have also, in addition to our research component, set up what we call a national facility. So this is shared infrastructure. This is very relevant for you because this has been essentially set up to serve the Italian community. So these national facilities serve a dual function, both as a research infrastructure and a training hub, and is meant to foster bidirectional exchange of knowledge from the Italian community to outside and vice versa. But essentially what the national facilities are high technological facilities that provide services that are accessible by Italian researchers through a grant application. So you will write a grant, and if your grant is successful, your experiment will not only be funded, but indeed done for you by our facilities. So this is really a unique model and a very empowering model to work. And now we have statistics. We have already approved hundreds of projects throughout Italy. The facilities perform genomics, not just standard genomic sequencing, but also there's a lot of multi-omic technologies, a lot of computational genomics, and ability to develop new protocols. We have a facility for genome engineering and disease models that, for instance, might take a cell and develop it into an ICA model, or indeed in an organoid model, and do gene editing, CRISPR, and validation. We have a computational data handling facility and really powerful imaging facilities based on light imaging and also in structural biology. Okay, so as I said, this is a facility that is available to universities, to RCS and public research entities. Access to projects is granted on the basis of scientific excellence and evaluated independently from us. So I'd really like to close with, first of all, an invite to visit us because we really want to become an opportunity to interface not just with my community, but with the broader clinical community. Very much encourage you to visit, to apply. We host exchange students and very much welcome the possibility for exchange. And just to conclude, I just really want to reiterate that, we are observing, or we are worldwide observing a real shift in our model, not just of healthcare, but also society that is based on genomics, not just in clinical application, as I told you, but also in driving, for instance, innovation in industry. We know the return on investment on genomics is huge, it's tenfold, and in the deployment of sophisticated approaches, such as, for instance, digital health system and artificial intelligence. This requires a change in mindset, and so we're working very hard with our legislators to change the public perception of the risks of genomics in Italy, and to hopefully come to a new set of ethics and law rules that allow us to work with that. And I'll just close by acknowledging all our scientists. This is my research center in our retreat in the Alps, and very much a welcome opportunity for exchange. And if you need any information or want to follow up on this, please just feel free to mail me. Thank you very much.
Thank you very much. Grazie, Professor Nicolo Soranzo. And it was really interesting to see the landscape of genomic science and even the future perspective that is much more integrated even with the new fields. So you have a real strong interdisciplinary, not multi, but interdisciplinary approach. You just try to lose the boundaries of each science branch. Okay, so now the session is open for discussion. We can continue to speak English, of course, but all right, yes. No, devi venire qua e ti devi presentare se devi farlo.
Okay, thank you so much, Professor Soranzo. It was really interesting for me, especially how you gave us a very good picture on what is going on in Italy. I come from Spain. I'm visiting a researcher in Sant'Anna. I'm a postdoc researcher in the University of the Basque Country, actually working for the Genome of Europe. But on the LC part, I'm a researcher on health law and bioethics. At some point, you mentioned the challenges of communicating individual findings to the clinic, from the research to the clinic. Very lately, I've been trying to carry out some work on this, on the ethical and legal part. And I wanted to ask you, because we know that with the, that's called the new sequencing techniques, and also with the data sharing that allows more, let's say, further research on the same data, we increase the possibilities of finding something useful for individuals. And you were especially talking on population-based cohorts, that we are talking about mostly healthy people. So my question indeed is, what are, in your view, in your experience, the main challenges, ethical challenges for the communication of individual findings on those population-based cohorts? Thank you.
Thank you. That's a really important topic. I think there are, I think two sets of really big problem or big important topics. One is around the biology, the clinical. What do these variants mean? How people perceive them. And the other one is about data security and protection, which is a huge, important but different topic. So with respect to the first, I think we're very used to start from the extreme of rare diseases, of clinical genetics or in newborn screening. Then the task is clear, which is that of diagnosing, right? And I think in that sense, already Italy has a problem because they are not doing genome sequencing, so they are only limited to the things that, screening things that we know already. What has become very clear that the moment you sequence a genome, that throws up a lot of information that you don't know how to understand, right? And you can take different approaches to that. The way we've been thinking about is that you need to make it very clear to people that participate to the studies that this is a research-only endeavor. So, there is typically the type of studies that we do, like UK Biobank, there is an explicit rule that there will be no return of incidental findings. Even things that are known to be potentially clinically important are not returned. And that's partly a pragmatic choice because it removes the burden of having to validate and counsel the people, but it's also, if you want, a philosophical choice that people will perhaps that create bias if people don't want to know, for instance, their genetic risk. So this is essentially one. But the problem remains is what do you do when you have all these variants of unknown significance? And I think population approaches, for instance, they use the development of these genome aggregation efforts at least helps you because it provides you with a baseline for how frequent the mutation is, what is the variance, of the penetrance of the mutation and expressivity. But it's not enough. That's each variant, potentially causative variant, will need to be validated through trio designs and segregation analysis. So I think it is very much an open field about how we want to communicate the risk and the uncertainty on the risk. And I think projects like Genome of Europe will at least help us understand the benchmark. What we found in the UK that really helps is that every project we do have an embedded patient PPI in a patient participant engagement, where essentially we have members of the public, essentially volunteers, that participate to the scientific steering boards of the project. So they are the ones that tell us what they're worried about. And so we can target a lot of the communication around this project to their needs and to their fears and expectations. And this is very much the kind of scenarios that will differ country to country, because different countries have different aversion to risk or different baseline of science. So for instance, in a single project, we're working both with UK Biobank, but also with a cohort of Bangladeshi and Pakistani that have high rates of homozygosity. And the attitude to risk is very, very different. You need to have personal engagement with them. So yeah, I guess there isn't a solution, but what is really important is that whatever strategy you take is tailored to that particular set of cultural and, for instance, even religious beliefs. So what Sangre had done at the time is to essentially issue surveys that were sent to people, asking them what they were worried about. So essentially a standardized set of questions that people could answer that would capture some of these cultural sensitivities. And the same questionnaire has been applied to the Italian population, and what came out is very different profiles. For instance, Italians tend to trust their doctor, but they don't trust the researcher. So that impacts also on how willing they are to share data to their own doctor. They're fine with that because it's typically a family doctor. But as soon as they hear that the data might go to human technopole, they get really worried, and let alone pharma. And so I think part of how you communicate and the kind of even the kind of data release policy is also informed by that. And I guess the other problem that we're encountering very much is related to the second problem, that of data sharing. And Italy again has a very conservative approach. The GDPR interpretation is as conservative as it can be. And so it'll be interesting to see now with new European legislation like the health data space, how this will be implemented. So we're trying very hard to build, this Molisani cohort was built because of the local connection, the epidemiologists with politicians, but we are struggling to get the same model to work, for instance, in Lombardy, because they are kept as a very separate entity. So I think in Italy, often personal relationship help build that trust that then help you take you to this type of groundbreaking applications.
Okay, so the ice is broken, and now the students can come to ask questions. Even curiosity, some aspects that are not clear, and maybe you want to ask to clarify some issue. Yes. Uno alla volta, c'è posto per tutti.
So, hi, I'm Sofia. I'm a first-year medical student. Thanks for the lecture. And I have a curiosity, so I have two questions. The first one is a curiosity about what you said before, about the high diversity of the Italian population, the genetic variability. So does this have some implications in the studies, in the research, in the genetic research? And the second question is about what are the most interesting populations to study for a genetic study, and what are the main targets?
Yeah, thank you. That's great questions. Okay, so I don't want to alarm anybody in the sense that, we know very well from population studies that the majority of differences are not between population, but between people, right? So we use this metric called FST that measure how genetic similarity between people. So we're mostly all this very similar to each other, but there are subtle patterns of differences. And so, for instance, in the study that I showed, what Alessandro was able to show is that if you take cities within Italy, they will have a different distribution of some key genetic variants. So in the grand scheme of this type of study, the impact, the clinical impact is probably going to be very little. But the problem can arise, for instance, say in a population, in a city in Italy, you have a high exposure. For instance, you have a pollution by heavy metals, I don't know, a raffinery or something, that creates an environmental risk. And so if in that city you also, by chance, have genetic variants that are enriched or depleted, you will think that those genetic variants cause cancer, say, but actually that's an environmental risk. So understanding these subtle differences in genetic structure is important because it allows you to have an evaluation of risk that is robust, without these spurious associations. So I think this is mostly what we're doing. And the place where this will make a big difference is in rare disease. Because of that, you have a genetic variant that has been, you think could be causal for a given disease, and you want to know exactly whether that genetic variant is shared by other people in that particular geographical area or not, because that will inform a prior that that's a potential causal variant or not. In terms of the extremes of these kind of subtle differences are populations that, due to their history, have been geographically isolated. So, for instance, Sardinia is one case, Finland is another case. Potentially, long-term genetic isolation creates essentially the enrichment of gene pool that might be associated with different diseases. For instance, Sardinia has a higher risk of multiple sclerosis and type 1 diabetes. This is well known, and this is due to founder effects, genetic effects. So going to your second question, which is what are the populations that are interesting or useful to study? These kind of genetic isolates, we call them, or special populations, are valuable because some disease risks are enriched compared to the general Italian population, so they are easier to find. For instance, there are variants that have been associated with cardiovascular disease that are 10 times more frequent in Sardinia or in Finland. Other types of, so these are kind of one category, where essentially their evolutionary history has created a unique genetic structure that is beneficial. We're using population of Pakistani and Bangladeshi origin, particularly the Pakistani have traditionally a culture of endogamous marriage, so they tend to marry within families, and that's a culture of practice. And we see it very much in London because the communities tend to be quite small. And so that creates, for instance, regions of the genome that are carried in homozygosis. So these populations are enriched for loss of function mutations, so we call human knockouts. And so this is also valuable because it allows you to see what is the downstream phenotypic consequence of knocking out a gene, for instance, in a person. And the third kind of element of this is really the idea that you want to make, there's a lot of human variation that is not present in Northern European samples, the one that have been sequenced until now. So we're doing a lot of efforts now to sequence population from all over the world to capture kind of predisposition that might be absent in Europe but present elsewhere. And this is really both a scientific reason to increase our knowledge of genetics, but also really a question of equity. We want to give every population the same benefits of potentially accessing genomic information for health. Thank you.
Great. So I have seen another volunteer. If you want to stand here, okay. If you want to go sit, okay too.
Good evening, I'm Ettore Feola, a first-year medical student, and during your presentation, discovering that different drugs have different efficiencies based on genetic variations piqued my interest, And I was wondering, is that principle being used clinically? If not, how far are we from clinical use? And secondly, is there a relation which could be used to determine the right dose of a drug based on genetics? Thank you.
Yeah, that's a great question. So I went through that part quickly, but this is really one of the key areas of innovation at the moment, and development is hugely important. So essentially, when I show you that chain of events, essentially that links a drug to a gene, to a genetic variant, to a disease risk factor, is really the main point of it. So the principle is that if a target, if a risk factor is on the causal path to disease, you can modulate that gene to treat disease or to make the disease worse, right? And so in the old model, pharma companies just relied on biology. They say, this gene is expressed in brain, perhaps I can try to target this gene to treat a brain condition. But they had no knowledge of whether that gene was actually implicated in the pathophysiology of that disease. So what we're doing now is we're using genetics to do what we call the in silico trials. So essentially, it's a principle called Mendelian randomization. You can maybe look in that review, but essentially, when you do a clinical trial, you typically randomize people in a placebo, in a treatment arm, right? And then all things being equal, you look at differences in effect of a drug in the placebo and in a treatment arm. The way we're using genetics, we say, let's imagine that this particular gene or risk factor that's targeted by the genetic variant is on the causal path to disease. Then you randomize people based on which genetic variant they carry, whether it's a risk variant or the other variant, and say, do I see a difference in outcome, in clinical outcome, based on the person having one allele or the other? And essentially use randomization of alleles at birth as a randomized clinical design. And this is the kind of principle behind then saying, okay, we now have genetic evidence, we take statistical tests, and we say, I predict that this drug is going to be efficacious for disease or not. And then you go and test it. And so, for instance, if you use this genetic approach, we could predict very clearly the modulation of HDL cholesterol has no effect on cardiovascular disease risk. And this was a clinical trial that wasted hundreds of millions of dollars, that we knew now with genetics, we know was never going to work. So we can use genetic evidence to point to those points of intervention that are more likely to succeed. But we can also use them to design what we call now intelligent trials, which essentially people are designed based on whether we think they're going to respond or not to a given drug. So some of the reasons for failure for clinical trials are not, their safety, but they're also efficacy, right? So say you have only the 30% of the people will respond to a drug, and we can develop a genetic marker that tells us which of those population, those 30% are the ones that are going to respond, then we can do a clinical trial targeting essentially those 30% and then increase efficacy for a subset of the population. So there are many, many different ways, but the point I want to make is that because we now have these huge databases, we can do all these experiments in silico, we don't need any more to go on, we just do the trials after. And so a lot of the trial design is done by that. Then the other point about, can you modulate a drug is also a very important one. We typically, the way we think about it is, and there are nice reviews that I can point you to, but essentially that, for instance, if you have that situation with a drug that you can modulate, a target, when we have multiple genetic variants that all act through the same genetic targets, we can build, we don't have a single time point, but we can build those response curves. So essentially, imagine you have a loss of function variant that disrupts completely the protein, but then you have a non-coding variant that changes, a non-synonymous variant that changes the drug a little bit, and then you have a protein coding, a non-protein coding variant that changes the expression even more. And so what we can do is to build those response curves. So we say, what is the correlation between the magnitude of a genetic effect on this target and the predicted response of the drug? And so this essentially allows us to understand what are the intervals of drug that are more likely or less likely to succeed in a clinical trial. And in fact, we can even go a step forward and say, okay, imagine that genetic variant is associated with increased risk of type 1 diabetes and decreased risk of multiple sclerosis, say, or celiac disease. And then we can also predict, so if I give the drug to treat celiac disease, am I going to increase the risk of type 1 diabetes? And so, again, these are kind of all genetic and statistical tricks, but they're incredibly, incredibly valuable. And when you add the multi-omic single-cellular, the things that the chance of success, predicted success, goes even through the roof, because immediately you can also go and look at environments through circulating protein, or you can look at, for instance, tissue specificity. And that drives also, for instance, understanding which cell you need to deliver the drug to. So I think you'll see that the reason why pharma companies are now investing hundreds of millions of dollars into this type of genomic research is because they see an economic benefit. And in fact, they have recruited the majority of the geneticists, of the generation that did this kind of studies are now all working for pharma because of this. So good new career opportunity as well.
Other question? Okay. If we ask a small question, we can have more questions. Okay.
Short answer.
Hi, I'm Giovanni Manasia, also from the first year of medicine. I thank you for the really interesting exposure, and I wrote my question because you never know. So, during the lecture, there have been showed the slides also about different allele distribution across far populations. And from the early beginning, it has been showed and underlined the strong connection between the expression of specific phenotype on the base of specific genome, and also the influence of the environment. So the question is, through all the data that have been collected, it has been thought to collect data also about possible migrations or long movements made by the individuals to investigate if changements into the environment from the native one can be crucial factors into the development of some diseases?
Yeah, so that's a great, very insightful question. Thank you. Yeah, I think this is exactly that. Part of what we are trying to do is to quantify what is the relative importance of genes and environments in condition. And we know even the heritability, so how much genetic component there is, depends on the environment, right? It depends on the environment. And so so it's hard to work with people migrating because sometimes these changes take effect. But what we are trying to do is to understand, take people, for instance, of the same ethnicity that are exposed to different environments. So, for instance, Bangladeshi that live in London and Bangladeshi that live in Bangladesh, right? Because, Bangladeshi living in London, they'll have the same genetic structure, but they'll have quite different environmental exposures. And then likewise, we can compare white British people that live in London and Bangladeshi that live in London. Again, they have very different exposures. So, for instance, in our study, in the single-cell study, the Bangladeshi and Pakistani have very high rates, not just of inbreeding, homozygosity, but also of, for instance, risk of cardiometabolic disease. They have a very high risk of diabetes, a very high risk of obesity, and this is very strange because they are much younger than UK Biobank. And this is clearly due to genetic differences, but also to lifestyle. We also see that they have a very high rate of allergic disease, and that's because they tend to live in highly polluted parts of London compared to wealthy middle class, right? And so essentially what we're using is these differences, to compare and contrast the information that comes from different studies to really understand what is the relative proportion. And I think one of the difficulties with this type of studies that particularly for environmental effects, you really need large sample size. You cannot do this study in 10 people or 100 people. You need large studies. And so, for instance, in Biobank, there's now 1 million studies on environments that look at things like the postcode. Where you live in Britain is a very good predictor of your socioeconomic status. And that, in turn, is predictor of your education level, or where you shop for your groceries, whether you eat vegetables or not, whether you exercise or not. So we now build in the databases that allow us to to make those inferences in a statistically robust way. Thank you.
Other question from the students? It is really interesting. If I took a picture of the audience from above, I will see a volcano plot. I don't know why, but it's quite strange. I don't know. I have a question from a professor, an engineer that followed your seminar. The name is Stefano Mazzoleni, and I just read the question. Good evening. Thanks a lot for the inspirational lecture to Professor Soranzo, and thanks for the organization of the event. I would like to ask if you envisage possible technological bottlenecks in the coming decades, for instance, as regards the NGS infrastructure, due to physical constraints in microprocessors, and if you consider quantum informatics as a possible technological opportunity as an early of omics science or machine learning, deep learning may mitigate these possible bottlenecks through robust predictive algorithms.
Yeah, no, we're already hitting the limit of the kind of ability to analyze the data that we have. So, in our single cell project, we generated data for 12 million cells, and we analyzed that against millions of data points in the human genome. So, at the moment, there is a lack, we are lucky that we work in an infrastructural institute with a good computational infrastructure, but for instance, this is a problem for many academic institutions. They don't have the computational firepower to analyze these data sets. So we are thinking, the way we're thinking about that is to build, the way it's been implemented in many places is to use cloud infrastructure for computing, but that creates problems of maintaining the data and the data secure. So this is a problem in Italy. We have some of the best computational infrastructures in the world. They're used by the physics community a lot, but they are not available to the biological community because they are not set up to protect personal data like genome data. So this is, that's a clear infrastructural bottleneck. So we're working with Cineca, with the Leonardo high-performance computing, but these are more legislative bottlenecks in how to access and make use of the infrastructure. On the other hand, while with the kind of analysis that I show you, the prediction are still very focused on individual problems, we are now essentially moving towards a model where we use artificial intelligence routinely. So we are using artificial intelligence to decode the parameters from imaging data. We are using artificial intelligence to put together data from this multi-omic or multi-layer data set. And again, some of the models we're hitting the limit of what we can train. So, for instance, we're training algorithms on 500,000 imaging data sets, and that's really computation intensive. So, I would say that we already use machine learning daily, and the question is how we make it more efficient. But I think the biggest hurdle is still where to keep the data and how to access it, and that's a problem.
So thanks to Professor Stefano Mazzoleni, Professor of Engineering at the University of Bari, but he comes from this school and is online. So other question from the audience? I would like to thank even friends that come from the Institute of Clinical Physiology of CNR, genetists, and even people that come from Scuola Normale that just arrived here today. Other question from the audience? Clinician? I have a question. So, it's really interesting that Molise, that is a small area of Italy, and follow a diet, a Mediterranean diet, has a so high risk, high cardiovascular risk. No? He tried to, discuss on the real effectiveness of the diet, okay? Because actually we know that med diet help to prevent cardiovascular disease, even to reduce significantly the risk of cardiovascular disorders. So I can elaborate this data in that community. How it depends on the absence of contamination, genetic contamination even.
Yeah, no, we don't know for sure. So what we are trying to do now is to build, this is partly why we want to go outside on this, right? Because we want to build a picture of Europe, of Italy. So we're working with another smaller cohort in South Tyrol, so in Bolzano, and one in Sardinia at the moment, because they already have data. And so we're really trying to understand to what extent the genetic risk is shared. Because if the genetic risk is shared, then we can infer the rest is environment, right? And we know that Molise, also population, of course, is aging, but they are also moving towards a more sedentary lifestyle where, people don't work, for instance, in agriculture as much as they did before. So the process is not, we don't have answers yet. I mean, I don't think it's necessarily a diet question. I think it's probably more a lifestyle, a movement. And this has been shown all over the world that that's the biggest determinant of increasing rates of obesity, as well as, consumption of processed foods. But this is partly why we need the other benchmarks, because, you can make that inference on a population.
Because the message is really strong. If you show that graph and people read the data, think that maybe the good lifestyle is not enough to prevent cardiovascular disease or to reduce the risk.
No, I think, so the Progetto Cuore that I compared to is a pan-Italian, is really truly representative of Italy. So, you'll have essentially, exactly that, people that live in urban environments together with people working, that live in agricultural environments or in rural environments. You'll have, heterogeneity in terms of, access to fresh food or education, whatever, and lifestyle. So, it's hard to make statements other than to see that we see differences, right, until we have found out. What we know from the kind of genetic exploration we've done so far is that we are broadly similar across, for at least the things we can measure that feed into this predictive algorithm, we are not that dissimilar. But it is very possible that there are even genetic variation that we have not measured that could contribute to these differences. So, in the case of Pakistan and Bangladesh, where of course it's easier to see these differences, it is very clear that the increased rates of obesity and type 2 diabetes are not environmental. They have a strong also genetic component. There is some risk that we might not yet have measured, but we know it's there. Yeah.
Okay. Question? Dr. Andreassi, do you have some question? No. No, because Dr. Andreassi is a genetist and is focusing even on aging and even aging induced.
Yeah. Nice. And perhaps the thing I can say is that, part of, the kind of stuff we're doing now is really, leveraging the things that we weren't doing before in Italy, right? Is large power, large data. But ultimately, it goes down to the task of finding genes, finding models, right? And testing those models, right? And I think part of the opportunities for collaboration and interaction is exactly that, is to say, do we come up with the likely targets from our studies that are worth testing in the lab? And perhaps we can use genetics to kind of drive some of the hypotheses, to inform some of the experimental work. So for instance, in the single-cell work that we've done a lot of work mapping RNA variation in immune cell types, so we can capture 30 main PBMC, peripheral blood cell types, and 65 different cell states, meaning these are cells that are, say, different types of B cells or monocytes that have different characteristics that make them, functional entities on their own. And what this type of studies are determining is that this is not just due to the expression of protein coding genes, but also regulatory genes. So we're talking a lot about microRNA. So we see a lot of cases where the genetic effect is mediated by a non-coding RNA that has a regulatory function on the protein coding and expression of protein coding. So it's quite hard to bridge the kind of thousands or hundreds of thousands of hits that we have from this type of studies to address them, but that's partly the challenge that we have as a community, right? Is, for me to sift through this data and come to you and say, this is really worth following up. And in a way, that's what also pharma do, they are doing. They're taking these leads and they put them into the preclinical development program very quickly, in a way that perhaps is even quicker than academia can do. But I think it's, perhaps some of the kind of mission of us geneticists is that to try and change the perception of, it could have been the perception of the early pharma studies that we already know the targets because we see the biology and we see differences. So what genetics allow you to do is to tell what is correlation and what is causation. And it allows you to focus on the causative effects. And I think that's really where the big gain of this type of approach is. It's that it drives biological insights from understanding causation. And this type of big data give you the statistical power to be able to be confident that that's causation and not correlation. I think that's perhaps the biggest lesson, that we learned is that like we design our clinical trials that are optimized, like we predict drugs, we can predict biology and causal biology, and that's very important. So, this is a challenge on us. We kind of spew out big data sets, but we don't often make a good job at making the data accessible and really for people to understand the value. And part of, working closely with, even within Human Technopole, there are experimental biologists that don't know how to use genetics, and part of what we're trying to do is to exactly, cross-fertilize on the utility.
Question? Okay, so if no other question are available, grazie molte per la disponibilità, per la bella lettura, grazie a voi per la partecipazione e come la professoressa Soranzo ha detto poc'anzi, questo back-to-back da Cambridge si concluderà il 23 maggio con la lezione di Sara Amalia Techman. Grazie.
Grazie a voi.
A questo punto c'è la fase finale, invito gli allievi a non fuggire via perché c'è la foto all'annuario e quindi lascio la parola agli allievi ma anche ai delegati degli studenti di medicina e chirurgia della Scuola Superiore Sant'Anna per questa parte molto interessante. No, invito la professoressa Soranzo a rimanere qui.
Innanzitutto grazie mille per la lezione che è stata molto interessante e molto chiara. E, sì, ora diciamo c'è un momento un po' rituale. Innanzitutto le vorremmo chiedere di scrivere un pensiero su quello che è un po' il diario che noi teniamo di Orizzonti, dove raccogliamo quelle appunto che sono i pensieri delle relatrici e dei relatori, non solo di questa edizione ma anche delle passate.
scritto in italiano, ho scritto: cari studenti, professori e personale di ricerca, è stata una bellissima opportunità di conoscere la vostra realtà di eccellenza e di condividere il nostro percorso scientifico con voi. Sono stata molto favorevolmente colpita dal livello di eccellenza e da curiosità dei vostri studenti. Il futuro è luminoso. Con affetto e ammirazione, Nicole Saranzo. Grazie mille.
Grazie mille. Poi le vorremmo dare questo, che è un attestato dove insomma la ringraziamo per essere stata docente per un giorno qui alla Scuola Sant'Anna.
Grazie mille.
E abbiamo anche dei gadget della scuola che si può portare.
Non si scorderà sicuramente di questa giornata.
Grazie a tutti veramente.
Grazie a lei.